Hi, and thank you for joining me. Firstly, I would just like to apologise for not being able to give this talk live, but hopefully this recording makes up for it somewhat. This talk is based on part of my PhD project, and it is focused on how we can improve frog conservation in fragmented and modified landscapes. Now, human-driven habitat loss, modification, and fragmentation is occurring faster now than ever before, causing species declines and extinctions. Now, within these fragmented habitats, one of the key considerations is how species movements will be impacted. Movements are really important because they are the key to genetic connectivity. If populations become isolated across space, they face an increased risk of becoming inbred and experiencing negative fitness consequences like reduced survival and slower development. Species also often have quite diverse needs across the landscape. They might breed, forage, and hibernate in different places, for example. And in fragmented landscapes, if species are prevented from reaching these resources, they may face an increased risk of decline. Naturally, we would expect habitat fragmentation to have quite a large impact on highly mobile species like birds or mammals. How it affects less mobile species like frogs, for example, is relatively less well known. When it comes to frogs, they're pretty small. You don't expect them to move very far, but population connectivity is really facilitated through large juvenile dispersals for a lot of frog species. So these dispersals can be as much as 5 kilometers in distance and 750 meters in elevation. So when it comes to assessing and managing the impacts of human activities on biodiversity, it's really important that we don't overlook these less mobile species. It is also important that we study frogs because frogs are in trouble. They are one of the most threatened groups of all vertebrates for a number of reasons. But one of the biggest threats to frogs is actually not listed here. One of the biggest threats to frogs is actually our lack of knowledge. So for a lot of frog species, for a lot of Australian frog species, we just don't really have much information on how they move across the landscape, let alone how they might be affected by habitat fragmentation. What is clear though is that different species, even those that appear quite similar, can sometimes respond in drastically different ways to habitat modification. So take the Burrelong frog and the Stony Creek frog, for example. These are two Australian stream-associated species. For starters, they're fairly closely related. They're both in the genus Latoria. They have very similar habitat preferences. Both of them occupy riparian habitat like this, often along the same stretches of stream. Physically, they're quite similar. They have quite similar life histories, and they do have overlapping geographic ranges, as you can see on this map here. So the Burrelong frog is there in blue. But despite all these similarities, they do have at least one key difference, and that is that Burrelong frogs are critically endangered under the IUCN. The numbers have declined dramatically since the 1980s. The Stony Creek frog, on the other hand, is considered least concern. So they're really common and they appear to be thriving in the very landscapes that Burrelong frogs are declining from. So of course that leads to the question of why? Now, I've been radio tracking these species and it seems like the Stony Creek frog is utilizing and moving across the landscape more than the Burrelong frog. Now, I've mentioned the importance of population connectivity for ensuring population resilience. So the question is, do differences in gene flow and genetic variation, is that what's responsible for these species' different responses? So my objectives were to assess and compare the genetic diversity and gene flow across both species, across fragmented landscapes, and also to determine whether the genetic structure of these two species is related to their threat status. So obviously I needed to collect some genetic samples, so I headed to the species shared range in the New South Wales Central Tablelands. Now, Burrelong frogs persist in several disjunct locations across the region, which is indicated roughly by the little pointers there. Some of these sites are fairly unmodified, some of these are quite modified, including sites next to forestry plantations and sites in agricultural and private land. So I sampled several sites across the region. I had collected over 120 genetic samples, and the plan is to analyse these using DartSeq. So DartSeq is a relatively new next-generation sequencing approach. It can provide thousands of markers across the genome. It's time and it's cost-effective, and it can detect population differentiation at small scales that are relevant to management. So overall, the outcomes of the project is hopefully to be able to identify genetically unique and vulnerable populations of Burralong frogs and Stony Creek frogs, to also understand species movements and habitat requirements, and to use this information to prioritise habitat that is important for species survival and improve their conservation management. So a huge thanks goes to the AWS and everyone who helped make this project possible. I'm very thankful for all of you.